Good morning. I think we got everybody. Good morning, good morning, good morning. Happy Tuesday. Whoop. Why did that happen?
Sometimes they start getting into that mindset of
All right, pause. Okay, good. I think we might only be missing one person. All right, good morning, everybody on camera. Good. How you doing? I like that hand in the air. There you go. All righty. How is everybody? All right. We are testing next, but we want to get, we started cell transport, so we're going to look at a couple of videos. I'm going to pull this worksheet up so that you can see where we're going with this. And I can get this to share properly. Okay. So. All right, I'm going to close that for a minute, but that's the worksheet we want. And I'm going to do this from student view. And I don't know why it comes up as a discussion, but it does. All right, so this one is in the module. So we want to go to, so everybody wants to go to your modules. It's not on the to-do list. Okay. And we are going to, let's start here. All right, so we're going to look at the cell membrane up close. So you can look at this one. This is a good video, but it's not as good as the one I'm going to show you. And then today's focus is on passive and active transport. And actually, we're just probably going to get as far as passive transport today. But the important part with this is that passive transport is the movement of molecules from high to low concentration without energy. All right, so that's like you sitting back in your chair and everything being done, and you didn't have to lift a finger. Okay, that's what they mean by passive. No energy is required. And then they have this word concentration gradient. All right, our concentration gradient is this expression up here. And this one, you're going to have to remember this. This is a rule. Things go from high to low concentration. So that is our concentration gradient is things going from high to low. Think of this, and we're going to be looking at molecules. We're going to be talking about things that are able to pass through the cell membrane, and what size molecules can pass without energy and what size molecules cannot. And I always like to say, think of your cell membrane as the screen on your window. And if you had a bucket of dirty water and you threw it at the window, most of the water would go through, maybe some of the teeny tiny dirt would go through, but a lot of things the screen would keep out. And that takes us to our word selectively permeable. Things can permeate. That means it can go through. So as I have down here, water and small molecules can get through the cell membrane, but because it is selectively permeable, it actually decides what it wants to go through the membrane and what it doesn't. So we're going to look at diffusion and osmosis today. I want you to have your little pen and paper out so that you can draw some pictures. because you're going to need it to help you do the worksheet. So like I said, today we're mostly going to focus on passive transport, and then we're going to get to active transport probably tomorrow, because we're going to look at these words endocytosis, exocytosis, and pumps. All right. So, and there's those words here. So do take a look at this page. But what we're going to focus on today is this worksheet. And again, this is part, we are doing an end of 9 weeks test, but it's going to include this. So consider this the review for that topic. All right. Okay, so we've got the first three columns up here. While we watch, so just have this downloaded and pushed to the side, and we'll come back to this after we watch the video. All right, so download that, put it to the side, and the videos that I have for you are on your playlist, so if you need to see them again, they will be there. But the first one we're going to look at is diffusion in general, or just how the behavior of the cell membrane works. And then we're going to look at pictures that I want to have you draw pictures so that you can answer those three top questions.
Bite Size Med. This video is on the different ways diffusion happens across the cell membrane. Transport across the cell membrane, it can happen in two ways, passive or active transport. Passive transport means it doesn't need extra metabolic energy like ATP, while active transport, it needs ATP.
Thing that you have to remember is in biology, ATP is our energy molecule. It actually stands for adenosine triphosphate. And that's the name given to the molecule that carries all the energy through our body. When later on we do respiration, we're going to talk about that a lot. But for now, just know that when they mention ATP, that is the energy required to get things to go back and forth.
Diffusion is a form of passive transport, and why it's passive is because the movement is along a concentration gradient. Across the cell membrane, diffusion can happen in two ways, simple and facilitated diffusion. In simple diffusion, the substance doesn't need help getting across the membrane, while in facilitated diffusion, it needs help, like from proteins. An example of this would be the carrier proteins. And we'll get to the different ways in a bit, but first let's see what diffusion is.
Okay, I'm gonna pause it there. So Raymond, as you watch this, some of the transport proteins are not for active. There are some that are for active, but right now we're going to talk about the facilitated ones, and they're not active, they're passive. All right.
To understand this, we take two solutions separated by a membrane. The solution has a solute and a solvent. So for example, in a solution of salt water, salt is the solute and water is the solvent. The concentration of the solute in the solvent is different in the two solutions. One is higher and one is lower. The difference between the two concentrations, that creates a concentration gradient. Now let's assume that the membrane is permeable to the solute, which means it's going to let the solute pass through. Molecules from both compartments have kinetic energy and keep moving around, bumping into each other and into the membrane. So the chances of them crossing the membrane will be higher when the concentration is higher. So the solute diffuses from higher to lower concentration until the concentrations of the two solutions are equal. This is equilibrium. So what drives them to move is the kinetic energy of the molecules themselves. No additional ATP was required. Since diffusion happens from higher to lower concentration, the movement is along or down the concentration gradient. Even at equilibrium, the molecules, they still keep moving around, but the concentrations of the two solutions, they stay equal.
All right, I'll pause that for a minute. Trying to go back and do a quick review. When you did in middle school, you did physical science or life science one, and you learned about solids, liquids, and gases. And the fact that all matter is always moving, even if it just moves slightly. So solids, something that is a solid has little molecules in it, and they're always vibrating, but they don't vibrate so much that they spread out all over the place. So that's what she's talking about. All the molecules have some energy, even if it's just a little bit of energy. So they're never really standing still. So that energy that she's talking about is the energy that the molecule has anyway. Okay. Um, a liquid, definition of a liquid is it has a little more energy so that the molecules kind of spread away from each other. And a gas, they have lots of energy. So they're wild and they're all over the place and they spread way out. Okay, so if you remember your definitions for solids, liquids, and gas.
There's no net diffusion that happens at equilibrium. Now, the same thing happens across the cell membrane. Substances diffuse into and out of the cell depending upon the concentration gradient from high to low. But there's a barrier in between the cell membrane. So for the substance to diffuse, they'll have to get through that membrane. The membrane is a lipid bilayer, so if it's something that's lipid soluble, like oxygen and carbon dioxide, it can diffuse across the membrane from high to low. So one of the factors that affects the rate of diffusion is lipid solubility. What else can affect it? The size of the concentration gradient, inside versus outside. If there's a bigger gradient, meaning there's a bigger difference between the two, the rate of diffusion will be higher. Smaller solutes again increase the rate of diffusion. But what if there's a thicker membrane? That means there's more distance to travel, which will slow down diffusion. Similar to that is surface area. Increasing the surface area would increase the rate of diffusion. So these are just some of the different factors that can affect the rate of diffusion in general. But what if the substance is water soluble and not lipid soluble? It means it can't get through that lipid bilayer, so it needs another path. And that's where proteins come in. So integral proteins, they can form channels. They can form pores, which are open at both ends for things like water. Now these are called aquaporins. These are water channels. They have a size limitation, so only water can get through. So for other water-soluble substances, integral proteins can form channels. Like for ions, like sodium, potassium, calcium, and chloride, they have ion channels. Again, they're selective, so they have a size restriction, but they also have a charge restriction. If the channel is lined with negative charge, it won't let a negative ion through it, even if it's small enough to fit. So they are size and charge selective, and they can also be gated. Very simply, that means they've got gates which could be open or closed. Open gates means ions can go through. Closed gates means they cannot, even if there's a gradient. So even if there's a concentration gradient or a difference on both sides of the membrane, if the ion channel is closed, the ions cannot pass through. But if the gates are open, they can move along the gradient from high to low. There are different types of gating, like voltage-gated channels, where the opening and closing of the channels would depend on a change in the membrane potential. This is like those voltage-gated sodium and potassium channels that we see working in the action potential. They could be ligand-gated, like with hormones or neurotransmitters, where after they bind, the channel opens and allows ions to pass. A classic example for this is the acetylcholine receptor on the motor end plate. When acetylcholine binds to its receptors, it opens the channel, allowing positive ions to move along their concentration gradient. Like sodium, it moves from high to low concentration. Channels could also be mechanically gated, like where pressure or stretch determine whether the gates open or not. So through open channels, when ions move, yes, they move along a gradient from high to low. They have a concentration gradient, so that's from higher to lower concentration. But since they are carrying charges, they can also have an electrical gradient. If one side of the membrane is positive and the other side is negative, a positive ion would move from more positivity to less positivity, and a negative ion would move from more negativity to lesser negativity. These electrochemical gradients are important when trying to understand so many things like the resting membrane potential, action potentials, and things like that. Now we saw how channel proteins work, but some substances need to be carried across the cell membrane, and they use proteins called carrier proteins. The carrier proteins, they transport substances from outside the cell to inside, or the other way around, depending on which way the concentration gradient is, but they still move from high to low concentration along the gradient, so it's not to be confused with active transport, where it goes from low to high. These carriers are used for substances like glucose, like the GLUT transporters that's facilitated diffusion. Amino acids also use carriers to move across the membrane. This kind of carrier-mediated transport, it can get saturated, which means after a while, as the concentration increases, all the binding sites, they fill up. So it can't transport anymore. The rate of transport has reached its maximum, and this is called the transport maximum. This kind of saturation wouldn't happen with simple diffusion because there's no carrier. And those are the different ways that diffusion can happen across the cell membrane. If this video helped you, give it a thumbs up. And
Okay, I wanted to go back just for 1 second. All right, so she mentioned, now in this case, what she's saying, it has to go against the gradient. The molecules are not going from high to low, but they're coming in because this is something the cell wants to have. So if it deems that this thing outside the cell needs to come in, even though we're not going from high to low, it has to physically bring it in. But it can only do that so much because there are sites. This carrier protein only has little pockets enough to carry in so much at a time. All right. But as far as concentration is concerned, it's going from low to high because there'll be more of them inside than outside. And that's because they want to bring it inside. It's usually something that they need to, for example, this is the glucose that she was talking about. We need the glucose to make all those ATPs. So we need to bring those as many of these molecules in as can possibly be carried by the carrier protein. Okay, and then there are some other larger proteins that need to come in as well. But they have to fit on these sites. They're very specific. They don't, if it doesn't fit, it's not bringing it in. Okay. All right. Now's the one I want you to draw. So have your little piece of paper out, and we are going to draw. All right.
Why waste another
Right,
summer not
notice I'm
loving
on
your
your
body?
playlist
With Space
in
44, we
YouTube,
turn
so I'm
that
taking
question
these.
into a successful business.
A woman has an
Welcome to Moo Moo Math and Science.
All right. So again, I want you to have a piece of paper out. There's going to be 3 cells that I want you to draw, and you're going to need to have those pictures so that you can answer the questions properly on the worksheet. All right, so everybody needs to be drawing. I'll pause it when we get to the 3
In
pictures.
this video, let's take a look at the difference between isotonic, hypertonic, and a hypotonic solution. Take a look at these red blood cells and the effect of different solutions.
All right, so these are the 3 pictures that I want you to draw, and I want you to draw the circle with the title at the top, and I want you to draw the little picture at the bottom. All right, so the first one is hypertonic. You've got some cells these are all red blood cells. And the first picture, they look all shriveled up and irregular. You don't have to draw that many. You can draw 2 or 3. Well, just do 3 or 4. 3 or 4 shriveled up looking cells or things that have an irregular shape. Down to the bottom, you want to draw 1, but it's important that you draw both arrows coming out of the cell and the H2O. And we all know H2O is water. The second picture is isotonic, and those are nice, pretty red blood cells. Those are how they're supposed to look. And underneath is a nice round circle, and you have 1 arrow where the water is going in, and you need to say H2O in, and then you put H2O inside the circle and to indicate H2O coming out. And then the last picture, those are also red blood cells, but now they're all puffed up and they're irregular shape, but they're puffy, and they even show one that's burst. So what we're having here is like a balloon. It got too much in it and they can't hold it anymore and it bursts. So that's hypotonic. And then the circle at the bottom, you want to show it where both arrows are going in with the water. All right, so I'll give you a few minutes to draw that. Again, everybody needs to be drawing because you need these pictures to answer the questions on the worksheet. And again, 4 circles inside your big circle is fine. You don't have to try to draw as many as you see in the picture. All right.
Let's figure out why sometimes the red blood cell stays the same, sometimes it shrinks, and other times it swells. To begin with, it'll be helpful to know the difference between a solvent and a solute. Let's look at coffee. The solvent is what the solute is dissolved into. The water is the solvent, and the cream, sugar, and coffee are the solutes. Now, in our example, we're going to use salt water. In the example, the blue circles is the solvent, water, and the white circles are the solute, salt. An isotonic solution is equal. Iso meaning equal, and tonic meaning strength. So the solute and the solvent will travel in and out of the cell equally, and the cell will stay the same. It's equal. Now, a hypertonic solution is different. Hyper meaning above, and tonic meaning strength. And notice that there are more salt or solute than solvent outside the cell. As a result, you have a higher concentration of solvent inside the cell. So the solvent travels out of the cell, and the cell will shrink. The solvent always travels from high concentration to low. Now, hypotonic, hypo meaning below, tonic meaning strength, and notice there are very little solute outside the cell, and a higher concentration of solvent outside the cell. So the solvent flows into the cell, remember high to low, and the cell will expand. I hope that's been helpful in knowing the difference between isotonic, hypertonic, and hypotonic. Thanks for watching.
All right, so again, hopefully you drew this picture, and we're going to take these pictures and we're going to compare it to the worksheets you have. There's one more video, but we're not going to get to that yet. We'll see how we do. But what I want you to look at right now is this worksheet. So inside your module, you have cell transport. Open up All right, so you see the table at the top? That is directly related to the pictures you just drew. You drew a picture with isotonic, hypotonic, and hypertonic. And all you have to do is it says put a check mark in the column that describes each one. So I'm going to do this with you. So I need you to answer me. So in the first column, we want to look at the description. If it's an isotonic solution, we want to look at our picture and what does the cell look like? Does the cell shrink? Is there no change to the shape of the cell? Causes osmosis, and remember, osmosis is simply diffusion of water. So to be honest with you, there's going to be a check mark in each of those boxes all the way across because we're talking osmosis for every cell. Or does it shrink? All right, so we got one check in the box for osmosis because we are talking about the movement of water. But where's the other check going to go? If it's an isotonic solution, which of those phrases describes a cell that is isotonic? And you can type it in the chat or you can unmute and just tell me. So look at your picture. Is it going to swell? Is it going to shrink? Or is it going to stay the same if it's isotonic? Thank you, Raymond. He said it's going to stay the same. So you want a check mark and doesn't change the shape of the cell. So you have two checks in the first column. All right, second column, hypotonic. So again, look at your paper. And we already have a check under causes osmosis because we are going to have movement of water. But is the cell going to swell? Is it going to stay the same or is it going to shrink? All right. Everybody agree with Raymond? Raymond's the only one talking to me this morning. Everybody agree? You can do a thumbs up. All right. So yes, so you're going to check the one that says causes the cell to swell. So in that column, you have two checks. One for osmosis and one for swell, which takes us to hypertonic. So again, we already have osmosis. And guess which one that's going to be? Is it going to swell, shrink, stay the same? That's your choice. It's going to shrink. All right, so you have so each of those columns should have two checks on it. So I'm helping you, everybody, that we just answered the whole first part of the worksheet. All right, the second part is vocabulary, and then based on what you just watched in the movie, you're gonna match those up. I want you to go back to the module for a minute, and I want you to look at this one. Okay, so let's look at this for a minute.
Observing diffusion. Substances move into and out of cells in a few ways. One method of movement is diffusion. Look at the picture below to examine a simple example of diffusion. What occurs over time as the molecules of purple dye interact with the water in the beaker? Hover over the picture to find out.
All right, the molecule.
Molecules are always in motion, random motion. The rate of diffusion is higher in gases than it would be in a liquid. The rate of diffusion is also higher with increased temperatures.
All right, so again, it moves. So you might have put the little drop in one spot and that made it concentrated in that spot. And those molecules want to go from concentrated area to an area that's less concentrated. So it keeps spreading out until it has no more room, no more places to go. It's getting in between all those water molecules, and then it's going to run out of little spaces between the water molecules, and then it'll stop.
Diffusion occurs down a concentration gradient. Diffusion is the net movement of a substance from an area of high concentration of that substance to one of lower concentration of that substance, or down a concentration gradient. Click the picture to see what happens to the molecules over time. You have probably experienced this in a room when someone arrives freshly doused in perfume or cologne. Those nearest the perfume smell it first. Then eventually the smell permeates the air in the entire room, reaching equilibrium.
All right, so this is what we were talking about with gases. Gases have a lot of energy. So you might have started off with all of the gases were concentrated in this little bottle. As soon as you hit the pump, they say, yay, I'm escaping. And they move as far away from each other as they can get, filling up the room. The definitions for solids, liquids, and gases, where solids have a definite shape and a definite volume. If you have a block of something, then no matter where you put that block, it's going to have the same shape and it's going to take up the same amount of space. With a liquid's definition is it has no definite shape, but it does have a definite volume, which means if I have a glass of water in a glass, then I have 8 ounces of fluid, and it's going to take the shape of the glass. If I pour that in a dish, it changes shape, but I still have 8 ounces of liquid. If I throw it on the floor, there's going to be 8 ounces of liquid on the floor, but it'll flatten out and take the shape of the no container in this case. All right. Gases have no definite shape, no definite volume. They take the shape of their container, and because they spread out, they take the volume of the container that they're in. So like I said, they started off with a volume inside that perfume bottle. As soon as you sprayed it, the volume now is whatever the room is, because that's how gases have so much energy. They just try to get as far away from each other as possible.
Diffusion across a cell membrane. Water, carbon dioxide, and oxygen are some of the simple molecules that will cross the plasma membrane by diffusion when a concentration gradient exists. They would be able to move between the phospholipids from one side to the other, as shown in the picture. The majority of the molecules move from higher to lower concentration, although there will be some that move from low to high. The overall, or net movement, is thus from high to low concentration. Eventually, if no energy is put into the system, the molecules will reach a state of equilibrium where they will be distributed equally throughout the system. Molecular movement is still occurring, even at equilibrium.
Okay, so you've got the picture, and it's in 3 stages. So the first on the left is showing that all the molecules are outside and there's no molecules inside. So those little blue dots are high concentration and they want to get to low concentration. So in the middle picture, you have some of the dots of, and you can even see some inside embedded in the membrane. They are passively passing through the membrane to get inside. This is intracellular space, meaning on the inside. Extracellular space means outside. All right, and it'll keep moving until it reaches equilibrium. So the last column is showing that there's an equal number of dots inside and outside. And what they showed in the other picture is sometimes it stops moving and sometimes there's an equal number going out as there is coming in. And it'll keep doing that, but sometimes it just stops because there's enough on both sides that there's no movement from high to low. Okay?
Carrier proteins facilitate diffusion. Some large or charged molecules cannot cross the membrane without proteins that help or facilitate diffusion. Carrier proteins embedded in the membrane serve as pores or channels, letting specific molecules pass. This movement of molecules down a concentration gradient by use of a transmembrane protein is called facilitated diffusion. The protein often changes shape, similar to a revolving door or a movable bridge.
All right, again, this is still passive transport. It's just that the and then the other video we watched kind of showed it a little better, is the molecules outside are specific. So there are different protein carriers that fit the molecules that they want to. So some of them are going to be round, some will be square, some will be whatever shape, but the protein carrier is designed to let in specific molecules. It's not letting everybody in. It's specific molecules. So this is still passive. It's just opening and closing, but it doesn't require much energy.
Osmosis is the diffusion of water across a semi-permeable membrane. Since osmosis is a special type of diffusion, water would move from areas where there is more water, or high water potential, to where there is less water, low water potential. In this diagram, the solutions in the beaker are separated by a semi-permeable membrane, which is represented by the red line. The membrane only allows the pink water to pass through and not the solute, which is represented by blue circles. Water potential is higher on the left-hand side of the beaker initially, compared to the right. The solute decreases water potential. Net water movement is from high water potential to low water potential until equilibrium is reached. The resulting volume in the beaker on the right may be different, but the overall percentage of water or water potential on both sides is the same.
Okay, so this picture always confuses everybody. Again, there's that red line represents a membrane, and that membrane is so fine that it does not let the little circular things, those are the solute. It's like the solute, but the water is the solvent. And notice there's water on both sides, but when we look at it, there is more pink on the left side than on the right. So this is what we're looking at. If there's more pink on the left side, that's a higher concentration. So it's going to go to the side that doesn't have a lot of pink showing. Okay, and it's going to keep on doing that. So it looks like the level of water went down on the left side because it flowed to the right side. But in all, what she's saying is there's a constant gradient. The amount of water in that beaker is the same. It's just now it's moved from one side of the gradient to the other. But now there's more pink on the right side. That's the water, and it's make those molecules spread out a little bit. Okay, but we still have, like, I think we had 7 of them on the left side. We still have 7 of them on the left side, but they're in less water now than they were before. So now this is the area of high concentration of salt, low concentration of water. So if it wanted to, it would turn and reverse and go back, because now we've got a lot of water on the right and a little bit of water on the left. So it could turn around and flow back the other way so that it can go from high to low.
Tonicity is the measure of how much water versus solute there is in a solution. If we can identify a solution as one of the following, we can compare it to another solution to predict the direction of osmosis. Hypotonic solutions are those with less solute. Hypertonic solutions are those in which more solute is present. Isotonic solutions have equal concentrations of substances. Their water potentials are equal, and water would move across the membrane in equal amounts.
All right, so that's the picture that goes with it, but it also applies to the cells, the red blood cells that you just drew, because those would be the cells in a solution.
When animal cells, such as red blood cells pictured here, are placed into each of these solutions, you will see the following results. In a hypertonic solution, there would be more solute and therefore less water outside the red blood cells. Water will move out of the cells, crenating or shrinking them. In an isotonic solution, there is the same amount of water and solute both inside and outside the cell. water would move across the membrane in equal amounts. In a hypotonic solution, there's less solute and therefore more water outside the red blood cells. Water will move into the cells, causing them to swell or possibly lyse.
All right, so that word lyse means that it'll burst or cut.
When plant cells are placed into each of these solutions, you will see the following results. In a hypertonic solution, there would be more solute and therefore less water outside the plant cell. Water will move out of the cells, causing the cell to plasmolyze, where the membrane pulls away from the plant cell wall. This will cause the plant to wilt. In an isotonic solution, there would be the same amount of water and solute both inside and outside the cell. Water would move across the membrane in equal amounts, and the plant cell is called flaccid. In a hypotonic solution, there is less solute and therefore more water outside the plant cells. Water will move into the cells, causing them to swell and become turgid. This would help the plant stand upright.
Okay, so we call this turgor pressure. All right, so you know your plants are doing okay if they're standing upright. There's a lot of, and I remember the cell wall is rigid, it doesn't move, but you have a cell membrane inside. So the first one they're showing you that the cell membrane will pull away as it shrinks. It's going to pull away from the cell wall, but the cell wall will still stay looking like a little box. And if you have a balance, the cell membrane is going to go back to touching the cell wall. And on the last one, it's going to actually kind of push against the cell wall, giving it firmness. And that's what causes the plant to stay upright. It's like their skeletal system. So it wilts, it gets weak and can't stand up if the water is flowing out. All right, so again, I want you to, and then we're going to look at these. Actually, don't look at that yet. We're going to get to that in a minute. So come back to your worksheet and use the rest of this time to answer the two sets of matching questions. And for this one, they simply want you to look at these words: diffusion, active transport, or equilibrium. So you're just going to write next to each picture or under each picture based on what the arrows are doing and what the substances inside and outside are doing. Are they diffusing? Is it something that is going to require active transport because they're big molecules? Or is it osmosis, or is the cell going to remain the same? So each of these circles represents a cell, and you're going to decide which of those words matches, and you can use the term more than once. Okay. All right, and then do this osmosis practice, and then you'll get as far as number 3. So try to get to number 3. And then we need, oh, you can get to number 4. But we're going to talk about these ones down here to the bottom tomorrow when we talk about endocytosis, exocytosis, and active transport. Okay. Okay. So use the rest of the time this morning to finish working on this part of the worksheet, the matching. So you're just taking the letters from up here and placing them on the line in both cases. And if you need to refer back to the module, go ahead and do that. And again, this worksheet is in the module. It is not on your to-do list. All right, while you do that, I'll do attendance. It's Tuesday. If you need extra help with something, please come at 1:30 or 1:35. Just log back into the Zoom. Yes, you may. All right, now you will, when we finish going over this, you will upload this worksheet uh, um, for a grade. And I don't expect this one to be late because we're pretty much doing this together in class. All right, so from time to time, some of y'all have connectivity issues, and I try to be paying attention to the list, but sometimes if I'm grading or something, you might be sitting a minute, so don't think I don't see you. It's just I'm not ignoring you. I do try to make sure that you get back in if you get kicked out. Not a problem. All right, so again, everybody's working on finishing the worksheet up to the osmosis practice. Try to get as far as number 4, and then we'll talk about 5, 6, and 7 tomorrow. Okay, that's what I was repeating. You're working on the worksheet. And you're going to try to answer as far down as try to get to the osmosis practice and get at least to number 4, but we'll check on that tomorrow. And we're definitely going to work on the bottom ones tomorrow. Those are active transport, and we'll talk about those tomorrow. No problem. Also, again, you're reminded that we are coming up on the 9 weeks, the end of the 9 weeks. Let me go back to your homepage. You had the exams schedule on your homepage, and this is 4th period. 4th period is the last exam. It's on Friday, October the 2nd. So it gives us a little more time to review. It also gives you up until Friday, October the 2nd, if you are missing any work. And a couple of you, and by a couple, I mean 2 people exactly, are missing the plant and animal cells from the Nearpod. So make sure that you go back and do that. Okay. For some reason, okay, I'm going to delete this one. There was another cell transport there that was worth 5 points as a discussion. It should not have been available. So I'm going to delete that. So Andre, you need, I know it looks like you submitted. I'm deleting that. So you need to resubmit it in the column that is available because that one should not have even have been available to you. That was a discussion. Oh, I know what this is. Okay, we're going to do this one later. And I didn't delete it because, let me see how I can adjust this. Okay, never mind, Andre. This one will be fine. I have to, this is a discussion question. And we'll look at this question as a class tomorrow. I have to make it so that everybody can see it. I don't even know how you got to see it. Okay. Okay, so I did change the dates on it, but let's make it available tomorrow. All right, I want to say to you, the gases can pass through the cell membrane. You don't need energy for a gas, any kind of gas, to pass through the cell membrane. Keep that in mind. Reassign. All right, Raymond, look at yours and then make some adjustments and then send it back. And remember, each column should have, at the top, should have 2 checks. All right, Andrew, I'm gonna let you look at yours too. So you should have yes in 2 places. And no in 2 places. So you should have yes in 2 places. And no in 2 places. So you should have yes 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 in 2 places. So you should in each of the columns at the top. And again, so Andrew, I'm going to send yours back for you to look at it too. And again, remember, gases can just flow through the membrane. Why does this say late? It's not late. What's the date on this? Jeez. Uh-huh. All right, so Andrew and Raymond, look at yours again and send it back. All right, and everybody else, hang on. Don't send them back today. Hang on until we look at active transport tomorrow. So complete as much as you can today. If you feel like you can answer those ones at the bottom, more power to you, but don't send it till tomorrow. All right, if you have no questions for me, you are free to log out today. And I'll see you tomorrow. Again, don't submit these today. Hang on to them till tomorrow.